We will just today talk about a new technology that is widely used to detect the molecular mutations or abnormalities within the genetic material. We have finished the cytogenetic techniques, the karyotyping, the comparative genome comparative hybridization and the FISH. All of these techniques that we already talked about are dealing with the chromosomal levels. Now, when we want to dig more and to, let's say, investigate the genetic mutations within the genes, or to focus more or zoom in the DNA strands, we have to find some technologies that will deal with the genetic material in this minute level. Well, as we know that Kary Mullis, he's the one who invented the PCR techniques in the 1983, and this invention was awarded the Nobel Prize after 10 years. The first publication was, let's say the application of the PCR to be published to the scientific committee, that the PCR was used to amplify the beta globin genomic sequence and the genetic material for sickle cell anemia. So they used the PCR as a technique to diagnose the sickle cell anemia. And therefore, this invention was, let's say, the change or the revolution in the molecular techniques. What's a PCR? PCR, from its name, it's the polymerase chain reaction. It's like a sequence of reactions that will happen in vitro to reassemble the DNA replication process that is happening inside the body. Well, why do we need this process? If we have like a segment of DNA, we need to amplify, or let's say diagnose this part of the DNA, or investigate more this part of the DNA. We need to conduct this technology or technique to amplify this certain piece of DNA instead of just copying the whole DNA. If you just relate the things, or let's say take an example of a book, and you just want to take a photocopy of, or you need only a small paragraph from the book. You don't just copy the whole book, you just copy that page, certain copy the number you need. It's the same concept, and instead of copying or amplifying the whole DNA, we'll just focus on certain segment or region within the DNA molecule or the DNA strand, and then you amplify it more, so you conduct the other tests upon this piece of DNA. So first of all, the technique needs a thermostable enzyme, which is here most of the cases, or the assays will utilize the Taq polymerase. The Taq polymerase is mainly an enzyme that was extracted from thermal bacteria, which is called Thermus aquaticus. It lives in the hot springs. So once they extract this type of enzyme, they will use it in the PCR assay. So it can resist the high heat. For example, the optimal activity of the enzyme here is 75 to 80 centigrades, and it can stand for more than this temperature. As you can see that here at 92.5, the half-life will be 2 hours, and once you increase the temperature, the half-life will be decreased. Like for example, here at 97.5, the half-life will be 9 minutes. It means that it's at high temperature, it will also be stable for a certain amount of time. And it has the ability to replicate 1,000 base pair strand of DNA in less than 10 seconds at the optimal, let's say, temperature. brief description for the PCR method that happens in vitro, while here you can see that this is the DNA replication that happens inside our body in vivo. You know that to start the replication, you will need a primase that will generate, or let's say synthesize the RNA primer, which is the starting point for the polymerase to come and extend this primer side. So the replication of the DNA and the synthesis of the newly stranded DNA will happen or will start after this primer. It's like localizing the place that the polymerase will come and react. This is the same concept for the PCR. Just like you will need primers, you will need some enzymes, you will need your template DNA, and so on. So what are the components of the PCR? As we said, you will need the DNA template, the one that is for our experiment, the one we already extracted in the lab, your human DNA. This is the DNA template. You will need some primers, and the primers will be designed according to your assay. Let's say you have a certain region of your DNA strand that you need to diagnose, or let's say a certain gene you need to detect. So the primers need to be designed according to the sequence. A forward and a reverse primer should be available in the assay, so it can, let's say, specify the location you need to copy. You need some nucleotides here, the ATPs, TTPs, GTPs, and CTPs. These are like the building blocks for the PCR. And for example, for sure, you need the Taq polymerase, the assay. All of the assay is working based on the efficiency of this enzyme, the polymerase enzymes. Once this is, it's a high fidelity, it's a thermostable, all the characteristics that is presented in the polymerase, the DNA, this enzyme, the polymerase enzyme, will be reflected in your assay. Let's say you need a PCR that will finish within less time than the traditional one, so you need to optimize your reaction based on the characteristics of the enzyme you will add. For example, there are some sort of rapid enzymes that will work and will add more than 1,000 that the base pair per 10 seconds, as the one we have mentioned before. It will add more within shorter time, so that the overall time will be reduced. You can, let's say, optimize the whole condition. You can change the tubes of the PCR instead of the regular one. You will add a thin wall so the temperature can get inside the tube very easily and fast. You can also use some thermocycler that has some developed heating blocks that will put the optimal temperature on time, and it will reduce the overall reaction. And for sure, you will need, for all of these components, you need to add a buffer that will maintain the pH and the optimal conditions for the whole PCR assay. So, as we know that the PCR is said to be exponential reaction. It means that once you start with a single copy, the copies will be increased, and the single copy will end according to the number cycle. For example, here we have 40 cycles, so it will be 2. You will start with 2 copies to the power 40, and you will end with this number of copies. So starting with 2 per cycle, you will see that the increment will be exponential. So you will end with this much of cycles of the copies of the first copies. And that's why the more cycle you have, the more amplified sequences as your assay need. Some assays, they just they are enough with 28 to 35 cycle, it depends on the, let's say, amount of DNA copies you are requiring. So if we want to visualize what happens actually in the PCR reaction, we need a primer, as we said, and it will be designed according to our assay. Let's say this is the blue one is the whole DNA strand. So you have to design your primer according to the region, to the flanking region that is uh, ahead from your fragment and behind at the end of your fragment. So you just specify the whole piece of DNA you need to amplify. You need a forward primer and a reverse one, and you need some DNA polymerase and the building blocks, which are the DNTPs. So according to what you design, you will get the amplification products or the newly synthesized DNA strands. There are main steps of the PCR, which are the denaturation, annealing, and extension. The denaturation is just simply the separation of the two strands of the DNA, because the polymerase, it needs to come and to do its work or do its job, or it's extending the or synthesizing the piece of DNA. So it needs the two strands need to be separated or denatured. When it is denatured, then your primer will come and anneal to the specific site of the primer annealing site, so the PCR can start. And after this, the extension step will be adding the nucleotide or the building blocks to the newly synthesized strand. So you will have the copied pieces as a result from the original DNA. These are just the steps of each step of the PCR technique, and how can you start with a target sequence of one copy, and then you end with this much of copies. As we said, it depends on the number of the cycles. So after 35 cycle, one copy of double helix will be copied 2 to the power 35 in this much number of copies. So it's considered as an exponential increase.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So as you saw in the video, that in each step of the PCR, it needs certain amount of temperature, so the process can start starting from the denaturation, the annealing, and the extension. So as you can see here, all the components of the PCR will be added to the thermal in one tube to the thermal cycler. So this is the automation of the PCR technique, which was a revolution to the whole molecular DNA community. So once you need to automate the process, you need to add all of these components to the PCR tube, and you conduct your reaction. You just put the or program the cycle. We will have this in the lab. So to start the PCR process, you need to just program the denaturation, annealing, and extension temperature according to your programs. Let's say here you will just need to set the temperature to 95 centigrade for a certain amount of time. Then you decrease it to 55, and then you again increase it to 72 for the extension step, and you stay like this until 35 cycle, for example, as a repetition for the whole process of the PCR. Then you have your final product or your amplicons. The automation also is developing these days, starting with the traditional ones, the PCRs, and ending to the rapid thermal cycler. As I told you, the heating blocks, the whole system is now focusing in having a PCR products in, let's say, shorter time with high efficiency. So according to this rule, you will see the developing machines, the developing even these, let's say, elements that is used to produce these types of thermal cyclers will be also modified, so you can have a better or optimal temperature in certain amount of time. So what's the components and details? First of all, we said we need primers. They are single-stranded oligos, usually from 20 to 30 nucleotide in length. And once you know the let's say the sequence of the strand you need to copy or amplify, you design your primers according to these sequences. So their length and their content will affect the PCR condition. Why the GC content is important over here? Because the melting temperature will be calculated according to these sequences. For example, here, if I want to calculate the melting temperature of certain enzyme, I just need to know what is the G and C content and what are the A and the T content. Then I multiply the GC content by 4 plus the AT content by 2. For example, here for this primer, you just need to count how many G and C nucleotide, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. You multiply it by 4 plus 2 by 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on. Okay, so you count the melting temperature, and according to your melting temperature, you will just, let's say, adjust your PCR program or cycle for the optimal annealing temperature for these primers. There are some artifacts. Artifacts means some, let's say, unexpected products from the PCR amplification. And this is as a result, for example, for the PCR mispriming. Mispriming means that once you have a primer, for example, this is your forward primer, and this is your template, and it should anneal exactly to the sequences as designed. But the mispriming, it means that some sequences will be skipped and it will not be let's say, align with your template, and therefore you will have non-specific bands, as you can see on the agarose gel. If you are running the traditional gel electrophoresis tanks, you will have some sort of extra bands, and they are non-specific, so you know that the primer has a non-specific binding site. And sometimes when you are just doing the capillary electrophoresis, instead of having, let's say, this is your expected peak, you might have some extra peaks because of the mispriming. And for example, on the other side, if you have let's say this is the gel, and you are expecting to have, for example, this band here and this band here and this band here, and because of the mispriming you will have some extra band over here. So this extra band is a result from the mispriming of the primers on the template. So if your target, for example, as you see here, is 150 base pair, the result will be 150. For example, if this is a 150, the result will be 150 plus 120 base pair. And same here, you will have one. It will not be here, it will be here because it's shorter. So it will be here, it's supposed to be on this side. So it will be shorter as an artifact. The DNA template, as we said, it should be the concentration that you will add to the amplification. It should be adjusted according to the 100 nanogram for cellular DNA. For example, if you are just amplifying viral DNA, you will need 10 to 100 nanogram. So the maximum is 100 nanogram. What will happen if you add more? You will have for the and the gel electrophoresis, if you want to visualize your result, it will be as a smear because it will be so concentrated. And if it's in the capillary electrophoresis, the peak will not be sharp enough so you can interpret the result. You will have some broad peaks, and this means, or you will have like extra peaks or pulled signals. All of these are because of the extra template or extra amount of concentration of template DNA. So you have to adjust it, and once you have, for example, high amount or high yield of DNA, extracted DNA, you just need to dilute it and you put it in the reaction. You will need also the DNTPs, as we said, and it should be extra, or let's say excess and in high amount, so you avoid the deficiency and the process. So you don't put the cycle to 35 and you add less amount of building blocks, so in the middle of the process it will end. So the amount of 0.1 to 0.5 millimolar concentration will be enough. So this is a result of the sum of the reactions. That is for example here, this is an enzymatic or PCR done for the diagnosis of fragile eggs. You can see here that the gel electrophoresis was run on 80 volt for 22 minutes, and the concentration of the agarose gel is 2%. Here are the wells, as you can see in the background, and these are the negative controls, and these are the patient samples. For example, here for this band, you can see that it looks like a smear, and this is because of the high amount of template DNA. It's more than 500 nanogram. And we said that the optimal one should be around 100 nanogram. So you see here it's like a clear band compared to this one. So we'll have a better results. The same thing for the capillary electrophoresis. The peaks will be clear, and you will be confirming your results because the amount of the template DNA was optimal compared to this one that looks like a smear. So here you can see that the template of DNA amount will affect the PCR reactions very clearly. Another component is the polymerase, and we said that the Taq polymerase is the thermostable enzyme, and it was a revolution for the whole PCR technique in the 86. There are some other types of the high fidelity enzymes or more efficient enzymes. Some of them have high temperature stability. Some of them they have, as I told you, that it will add more or amplify more sequences within the specific range of time. Here it says that the minute amount or tiny amount of polymerase will be enough for the whole cycles, because it will just let's say work. It will just add the nucleotides. It will not be consumed like the uh, DNTPs. The DNTPs, once it's attached or it's used for the extension step, then it will be reduced and it will be consumed. The DNA polymerase will not be consumed because it's like the working men, they will be there compared to the blocks that they are building with. The blocks will finish, but the men will be there, but their efficiency will be reduced with time, so they will get tired. It's the same here for the enzymes. After a certain time, it will be reduced in activity, and the efficiency will just be reduced. But tiny amount will be more than enough for the whole PCR cycles. Here, for example, they are using the PFU turbo enzyme again for detecting or diagnosing the fragile eggs. As you can see here, the fragile egg syndrome is like you will just detect a triplet repeats, the CGG, and this assay is used to detect this deficiency within the molecular level. Here, just letting you knowing that the enzyme is used is not the tag, it is just the PFU turbo. For the assay was run the gel for 80 volt for 50 minutes, and these bands was a result for the assay. Another component is the buffer. The buffer, we said, it's just to maintain the optimal pH or optimal conditions for the reactions. Just instead of having the in vivo DNA replication with its conditions, we just have to make the same conditions in vitro. For example, here the pH levels has to be maintained in an optimal range using the HCl. The sum of monovalent cations and the KCM and ammonium sulfate are also important for the enzyme activity, and again the MgCl2 or the magnesium amount or ions is also required to have optimal enzyme activity. Some of the non-specific PCR amplification products might occur, like, for example, the one we have previously mentioned with the mispriming process. Why does this happen? For example, the annealing temperature is too low, so you have to increase your annealing temperature. You might need a hot start. You added too much magnesium, so the misannealing is there or the mispriming is there, so you have to reduce the amount of magnesium. You have to design more specific primers so they can exactly anneal to your annealing sites. Sometimes you have extra product because of too much DNTPs, too much polymerase, the timing is not adjusted, or the number of cycle is high, so you have to reduce it. And finally, if you have some template contamination, it will result in also extra products that are not really the case of the used or the original template. It will just be a contaminated DNA strands that will appear as non-specific products in your gel. Why or when do I use the PCR? Sometimes you need to diagnose some genetic, let's say, diseases or disorders, like the fragile X example we have, the sickle cell anemia, or any other molecular level diseases. You will just need to conduct a PCR and then a gel electrophoresis to visualize the products. Sometimes you need to test for some mutagens, you conduct some DNA cloning processes, polymorphic genetic markers, which is mainly used in the forensic genetics to just uh, detect the short tandem repeats that will differ between individuals, so you can conduct the DNA profiling. Sometimes you need to detect the mutations or even to conduct the DNA sequence process. What are the advantages? It is rapid and easy to use. So within few hours you will finish. And we said you can optimize your reaction, so you can also decrease the time to even 1 or even 30 minutes in some of the publications. It is sensitive, and because it is so sensitive, the disadvantage is it is highly prone to contamination and inhibition. That's why you have to pay a clear, or let's say high attention, or high contamination-free steps, so you don't have extra or unwanted products. And for some sort of difficult sample, the PCR is very good option, because you will just, as we said, it depends on the site you are looking for. So once you extract your DNA and the extract is good, then you will need to conduct the PCR to amplify that region. On the other hand, the disadvantage will be that you need to know the target DNA sequence. So it's not something global that you will add anything to the PCR and it will just detect what you want. No, you need to know the sequence exactly. So as we said, you need to design the primers, the forward and the reverse primers based on this sequence. So one of the disadvantages that the sequences that you want to detect has to be known from the beginning. The size for the process is limited, maximum of 5 kilobase. So after the 5 kilobase, you don't have any product. Sometimes you will have copying errors because of the mispriming, for example, because of the non-optimized components of the PCR. And as you said, because it's sensitive, you will have contamination and inhibition. The contamination will be, let's say, a result from contaminated template DNA, contaminated surfaces you will work at, and even contaminated tools you are working with. Let's say the pipettes, you use the pipettes in a certain area, and then you piped your PCR component with that, so you will carry on some contamination like the extra template DNA or any bacterial contamination. The inhibition will be as a result of the type, for example, of the sample you are extracting. For example, if it's a blood sample, then it will have the heme. The heme molecule will just inhibit the PCR. It will just incorporate to the primers, and it will affect its any link to the strands. That's why it will inhibit and affect the reaction of the PCR. Sometimes the bone, the calcium in the bone, the soil, all of these depends on the type of the sample you are entering the PCR. You need to make sure that your extracts are free of these inhibitors. So to design your PCR lab, you have to make your lab unidirectional. Unidirectional, it means that once you go through unit per unit, you don't go back again, so you don't carry out any extra amount of DNA or you cause any cross contamination. So you have to design the pre-PCR area and the post-PCR area separately. Some labs, they just make the post-PCR as chambers or hood, and it's highly or totally separated. And there are some filters, so you don't carry back your contaminated and amplified DNA. To decontaminate the surfaces, you need the UV light. to degrade the amount of the DNAs that are available. You need some bleach, 10% bleach, to just get rid of the degraded DNA or any DNA that is on the surface. You need 70% ethanol or any type of alcohol, so you make sure that any spores or any bacterial contamination is gone. And you need a blank control to be carried through your assay, so you make sure that there is no contamination. Once you see the blank control is free of bands at the end of the gel electrophoresis, you will confirm that your assay is free of contamination. So you need always to add a blank control to your PCR. Why you don't need to, let's say, carry back your tools and use the same tools in the PCR and the pre-PCR? First of all, the pre-PCR is where you process your sample, you store your sample, you extract your DNA. These are all considered as the PCR, pre-PCR. And then it comes the PCR step where you conduct the PCR techniques, you add your component, you have your thermal cycler, and after that, the post-PCR will be the area where you make the gel and prepare for the gel electrophoresis. So once you come here, you will have million and billion copies of the amplified DNA. So let's just imagine that you carried one drop of the product here back to the PCR, you will re-amplify, and you can imagine how many copies you will result in. That's why you have to separate this area now from the other one. Some labs, they also use color categories. Let's say the pre-PCR, they will use certain color. And even if you notice that the tools also and the disposal containers also, they have the same color. So once you see a green color, let's say here in the post-PCR, you know it is here. So you have to decontaminate and then you have to bring it back. So these color scheme or color categories is very useful, especially in the PCR working stations. And as you can see, even the lab coat has to be colored schemed. For example, here, the lab coat, the pipettes, the gloves even, the safety glasses has to be specified for certain stages. The benches or the surfaces where you will work has to be contamination free. You have to clean with bleach, as we said, then you have to decontaminate with the 70% alcohol, so you make sure that you are just preparing the required DNA template and it's not contaminated. And it should be in one direction, starting from the green as you see the arrows and ending with the red unit. There is what's called a multiplex PCR. A multiplex PCR is having many PCRs in one tube or one reaction. Let's say we designed a set of primers forward and reverse. For example, here let's just, if I have like this strand of DNA, I need to amplify this region, and this region. At the same time, instead of having this separately, and this separately in one reaction, in three different reactions, I just design my primers. I'll just need a forward and reverse primer for each location or each site that I need to amplify or detect. So here, for example, I will have the 3 sets or 6 primers forward and reverse for the whole, for the 3 locations. I'll just add everything, the component, and I will just adjust my component and add them in one PCR tube. Everything will be added in one PCR tube, and in the result, you will just see your band clearly as it should be. Okay, so after you finish, you will see this is the result, the gel electrophoresis having each locus as you designed your primer accordingly. So this is what we talked about. For example, here if I have 3 strains I need to detect, I just add all the 4 primers in one reaction in a single tube, then I analyze them. And once I conduct the PCR, I conduct the gel electrophoresis after that, and I compare my results. And you can see that all the strains are available according to the dyes that are previously. For example, this is here the DMD detection or diagnosis uh, system that it says that the DMD is a recessive disorder, X-linked recessive disorder that is caused by exon deletions. So you expect to see any deletion in these sets of exons. So once, for example, here, if you want to detect, let's say, this 47 exon, you just go and look around and you see, for example, this is here deleted. So you know which exon is deleted, and therefore you can relate it to the certain molecular disease you have. Here it just says that the chromosome X, because it's an X-linked recessive disorder, the gene will be located in X on the P21.2 arm. Once you magnify more, you will see that it is 2.22 megabase with 79 exons. So the deletion or the hotspot for causing the DMD disease will be the deletion in the exon 40 to 54. So once you see this deleted in the previous slide, you will know that the person has the DMD disorder. If you want to explore more, you just go to this virtual lab and you will have and practice more PCR techniques.
PCR, or what we call it quantitative real-time PCR, qPCR. And the concept is the same concept of the traditional one, except that the detection of the target sequence will be reported at the same time. It's not like the traditional one. You will just perform your PCR and then you will wait until you detect it on the gel electrophoresis. No, it's just you will design your primers and it will be detected at the same time using the fluorescent dye, with no need for post-PCR procedure, which are the gel electrophoresis or the capillary electrophoresis. It helps us to have the quantity of the original template, so we design a specific prop to target these sequences, and then we can know the quantity of these targeted sequences. The steps of the real-time PCR using the same PCR machine is the annealing of primers. Once the elongation will happen using the polymerase, and once it attach or reach to the reporter or the signal, you will see a fluorescent signal reported. Like now, you see the enzyme is working. Then once it's reached the reporter, the signal will be detected as electric, let's say, peak or signals. You need to have some housekeeping genes in the assay. It is just a reference nucleic acid that is quantified to the DNA of at the same time with the DNA of interest, so you can know that your assay is working good and you minimize the errors of the quantification. Some examples are the beta-actin, cyclophilin, and ribosomal RNA. So if we will just look at the real-time PCR, we can compare it as the bacterial growth curve. If you know the bacterial growth curve, like you start with a lag phase when it's constant, then suddenly it goes to the exponential phase, and finally it will reach to the stationary phase according to the number of cells or the growth of the bacteria. The real-time PCR, it's the same. You will have a background, which is the lag phase, then the exponential phase, which is the log phase, and finally the plateau where it is stationary or it is stable. You can figure out the threshold cycle, which is the CT, when it is meeting with the curve of the log will just start to go to the exponential phase and finishing the lag phase. Another curve, so just to make sure that the concept is clear, the cycle numbers, the more the cycle numbers, you will see some fluorescent intensity according to the reporter signal that you will have once the polymerase will reach the reporter. So here you will see kinetic detection. So here you can just quantify your DNA, and here it is the end of the PCR when it goes to the stationary. And this is, for example, your sample compared for the standards. As we said, that you have to know what's called a threshold. A threshold is this dotted line, which is the value of unspecific fluorescence before specific amplification of the target. So you have some non-specific fluorescence, then once your target starts to fluorescent, you will have this exponential. So the threshold is these values of the unspecific fluorescence. The CT or the cycle threshold is the PCR amplification cycle in which the reporter fluorescence exceeds the chosen threshold above the background fluorescence. It means that this is the cycle where the first reporter will report its signal, and you can start detect your template DNA. So how can you figure out what is the CT value for your sample? You just have a baseline for the non-specific fluorescence. Then you have the beginning of your exponential phase. You just take the reading when it is intercept with the baseline. So the CT here, you just take this line and you go down and you figure out which cycle number it is, then you say that the CT is, for example, 30 cycle and so on. The fluorescence signal, as they say, will reach the exponential growth in early cycle when lot of target is present. It means that when you have your curve and the CT is high, it means the CT is low, sorry, so it means that you have lots of target is present. It means that you don't need lots of cycles to detect your target because it's presented in high amount. So once it is started, the cycle is in the early cycle phase, then you will have your target. While when you wait for more cycles, let's say here it is, for example, it's here uh, 16, for example, compared compared to the other side, for example, here 16 and here, for example, 30. It means here it reached to later cycle with 30 cycles, so you can barely detect your fluorescence or your template. Here it started with 16, you got your signal with 16 cycle. It means that you have lots of target of DNA, so you don't need more cycle to see what is there in your template. So this is how you read it. The more the cycle number, the less amount of your DNA is presented. You have to sure use some standards, positive and negative samples and a standard positive standards, so you can know that these amount of standards are already known, so you can detect the, let's say, approximate number of a quantification of your sample compared to these standard. The fluorescent dye in the real-time PCR they used before to use the ethidium bromide, but now because it is carcinogenic, they they used some non-toxic dyes, which are the cyber green, for example, or sequence of non-specific qPCR. These are like the fluorescent that are used instead of the carcinogenic ethidium bromide. Uh, we have two type of fluorescence system that is widely used in the qPCR. We have the cyber green and we have the TACMAN. The cyber green is considered as chelating agent. Chelating agent means that it will interfere with the sequence of the target, and that's why it will be reported as a signal of fluorescence, while the TACMAN is just a fluorescent label specific probes, where you design a probe with a specific labeling and you wait for the polymerase to act, and then the reporter will be just reported as a signal fluorescence. So this is the cyber green, where you can see the role of the intercalating agent. When there is a double strand DNA, the cyber green will bind the minor groove. If you remember the minor groove and the major groove of the DNA, so it will just bind to the minor groove, and then you will see these cyber green dyes as it is intercalating to the DNA. So once it's bind, it doesn't bind to the single stranded DNA, so it will not report any مثلا, for example, the RNA. It will bind to your double strand, so this is the targeted DNA. It will enter the assay as double strand, and it will interfere, as you can see, inside the double stranded DNA. So once you have a primer, the primer will just attach to its specific location, and the PCR will start. And once it reached to the, let's say here, it reached to the dye, it will report. These red arrows is reporting the signal. So the emission signals will be reported as, let's say, signals to measure the quantification, the quantity of the DNA. So the more you go and extend the strand, the DNA strand, the more signal you will have, and then you will know the amount of your DNA. So as you can see here, this is a qPCR assay using a cyber green with different sample. These colors are reporting different sample of the amount of DNA. As you can see here, it is 25 nanogram. It means that this is in the 4 starting here as the more amount. It has more amount of DNA. Once you go further, you will increase the cycle. It means that you have less amount of target DNA compared to the signals you got from the beginning of the cycles. It means here the more target DNA, and once you go further cycles, it means you need more cycles to detect your DNA because it is not presented in a high amount. So here you will have a less amount of DNA template. The other type of the fluorescence system is the TACMAN system. As we said, you will need a reporter, and there is a quencher. The reporter will attach to the 5' side. It will covalently band to the 5' side, and the quencher will be added on the other side. So it's just like pulling down the reporter. So once it reached here to the polymerase will reach to the reporter, this one will just quench it and it will be reported as a signal. So this mainly used in the quantitative PCR and microbiology and oncology, among others. So the main, or let's say the most used system will be AttackMan compared to the CyberGreen that we previously talked about. So again, this is how it works. The polymerization will happen and the extension will happen. Here you have the reporter and the quencher of the probes. You have a forward primer and reverse primer. Once you see that the extension is happening and the signal is reached, and the polymerase reached the reporter, it will be reported or cleaved and reported as a signal. Then the quencher will just allow the attachment of the probe from the strand, the target DNA. Here it's just, let's say, explain what's a probe and primer bind target, which is FRET. How does it work? It's the same concept of the tag polymerase, where it describes the energy transfer between two light-sensitive molecules, which is here. Once there is some light resource on the reporter, it will just transfer the energy to the quencher, and therefore they will be just detach and emit their fluorescence, and the accumulation of the signal will be reported as spectrum or some peaks that will detect the quantity of the target DNA. So here again, if we want to see the amount of DNA, you can see here the least cycles have the more amount of DNA. So here, if we compare 15 to 30, for example, here it has more amount of DNA compared to these at the end. And when you see that the curve is like going closer to the threshold, it means that this is the least amount of DNA you have. If you see here, it says here to the power 0, to the power 1, and once you go here, it is to the power 5. It means here it has the more amount of the DNA. And if you see the standard curve, you will see it will go this way because it will start high, then it will just go low until it reached the baseline. Again, in the real-time PCR, we can use what's called a multiplex qPCR. A multiplex, as we always say, that it is many assays that is happening in one reaction. So, for example, here I have a forward and reverse primer, and I have a probe, the same concept, but here I will just design two different props with many dyes as a reporter. For example, here the reporter here is purple color, here it is Vic dye, here it is Fab dye, and so on. So as you can see that changing the reporter dye will just give us many signals according to the detection of these fluorescence colors, and it will be detected as different colors, so it will be used for for good use to be interfering in the assay of qPCR, and it will be reported as different colors and different peaks. Therefore, you can quantify many alleles or many locations at the same time. So if we will compare the CyberGreen to the TACMAN probe, there are some advantages and disadvantages. One of them, the CyberGreen is low cost and more suitable to relative qPCR. The tag probe we can perform the multiplex qPCR as we said, and this is a strong point or strong advantage about the tag pan because the multiplex will always reduce the required time for the assay. It will detect more loci or more alleles at the same time. Instead of just repeating the assay 2, 3, 4 times, you will just do the same reaction in one tube and you will end up with uh, conclusive results. The TACMAN also has a stronger specificity, so it is specific to the target DNA you are looking for because you will design your probes according to the target, and once it's reached the probe it will be reported, while here it is just not that specific. It will just work on the intercalating ability of the dye to the DNA strands. The disadvantages: the TACMAN has a high cost and it is complex operation. As long as it's complex, it will cost more. While here, the disadvantage has low accuracy. It's not that accurate, and as we said, compared to the TACMAN, the specificity is low. And we can't perform a multiplex PCR using a cybergreen because it is only one type of dyes that will be used as intercalating agent, and you can't just use different dyes. Like here we can use the Vic and the FAM and so on. Here it is just one type of dye, so it is not applicable for the multiplex PCR. This is the machine for the real-time PCR or the qPCR. As you can see, it's just a 96 well plate. You will put it in the block and you will just close it, and it's attached to a monitor. You will enter your it's like a table. You will enter your sample names and location in the plate, so it will give you for each location. It will just detect the signal and it will give you as peaks, as we said, or a curve, so you can know the CT, and according to the CT you will know that your target is presented or not.